Okay, we can start, Dr. Tariq, please.
Okay.
Go ahead.
Bismillah ar-Rahman ar-Rahim. In the name of God, the merciful, the compassionate. Assalamu alaikum wa rahmatullah. Good evening for attendees who joined us from the Middle East and Europe, and good afternoon for attendees who joined us from USA and Canada. My name is Dr. Tariq Azzuddin, Research Institutional Official, Consultant and Medical Program Director, and I am pleased to be your host today. After the overwhelming attendance and the standing ovation we received for the previous webinar titled How to Present a Successful Proposal and the IRB, we were encouraged to carry on with other exciting topics for research and researchers. These webinars are the fruits of a dedicated teamwork and efforts from the Dammam Medical Complex targeting research and researchers under the care and patronage of the CEO of the Dammam Medical Complex, Dr. Zakaria Safran, and organized by the Training and Academic Affairs under the leadership of Dr. Malik Al-Muteri, while the logistics were professionally executed by the event group organization team. I wish now to give you all a little introduction about the webinar series. Today, we are pleased to start the first webinar of a series of webinars accommodating all the modules of the Good Clinical Practice, abbreviated for GCP. Attending all the 7 webinars and successfully completing the quiz at the end of the 7th and last webinar will qualify you to receive the certificate of completion of good clinical practice and with 7 internationally verifiable CME, or also called CPD. I wish to inform the audience that everyone can feel free to post his question using Twitter hashtag research_Saudi Vision 2030, and we will take them in order after the lecture to the presenter. And if there will be an excess of questions than the time frame permits, the presenter will kindly answer them later in Twitter. Regarding the audio icon, it is located down to your left of the screens where you can easily unmute it. This GCP course is an essential international ethical and scientific quality standard for clinical trials. It also serves to protect the rights, integrity, and confidentiality of any trial subjects. Today, our first webinar will be titled Introduction and Principles for GCP. To present this exciting and essential topic, we are thrilled to invite Mr. Naeem Noordin, who is the CEO and Managing Director for SIARA Limited UK. Mr. Naeem is a graduated in pharmacology with toxicology from King's College, University London, where he started his career as a clinical pharmacokineticist. During his 20-year career, Mr. Naeem has held various positions of increasing seniority and is actually the CEO of SIARA Limited UK, which he founded in 2007. His company, highly specialized and focused organization offering training, development, and consultancy in all aspects of international council for harmonization and good clinical practice and related guidelines, as well as for personal effectiveness and behavioral skills. Mr. Naeem brings with him over 20 years of experience in clinical development, training, consultancy, and education spanning Europe, United States, Middle East, and Asia. Mr. Naeem is also a lecturer at Cranfield University, UK, and a certified facilitator and behavioral skills trainer. Mr. Naeem has worked extensively in the Middle East and Asia, developing and building clinical research capabilities with the regions. In Saudi Arabia, Mr. Naeem was intimately involved and chosen to establish the structure of the Ministry of Health Ethics Committees and was heavily involved in developing the standard operating procedures and deploying the training for the Ethics Committee members nationally. Please welcome Mr. Naeem Noordin.
Thank you very much, Dr. Tariq, and good evening, everybody. And it's a real pleasure for me to be here and spend the next few evenings with you every day. And a very warm welcome, a very, very warm good evening from the UK. And also a very big thank you to Dr. Tariq for providing me with the opportunity again to speak with you this time. So what I'll do now is I'm going to go straight into the lecture, and I shall just share my screen with everyone. Perfect. And everybody can see the screens? Yeah.
Yes.
Okay. So what I'm going to do over the next hour or so is give you an introduction to and the principles of good clinical practice. So what I have as the objectives for this hour or so is to give you an understanding of the history of good clinical practice, understand the purpose of good clinical practice, very importantly, understand what are the principles of good clinical practice, and then to finish off, I shall provide you with an overview of drug development. As Dr. Tariq said, I shall spend an hour talking about this, and at the end of the hour, I shall take any questions you have and address them for you. So first question is, we hear good clinical practice. What is good clinical practice? How did it become to what it is today? I'm going to discuss now. So good clinical practice. Here is the definition of what good clinical practice is. Good clinical practice, it's an international ethical standard for the international ethical standard and scientific quality standard for designing, conducting, recording, and reporting trials that are going to involve the participation of human subjects. Very important words over here are international and it's ethical. Ethical, scientific. These are the foundations of when we're going to do research. It's a standard, an international standard that whenever we're going to do research anywhere in the world, because it is international, we shall follow this standard. So GCP, an international ethical quality standard. So we have this, but what is the purpose of this? What will good clinical practice actually achieve? If we follow an ethical scientific standard, what are we trying to do? Well, it's simple. We are trying to achieve two things whenever we do any type of research. If we follow good clinical practice, we will ensure that the rights, the safety, and well-being of clinical trial subjects are protected. So ethics, putting the subjects first, protecting the subjects in a clinical trial, protecting the subjects in research. Now, I'm going to use some international terminology. We usually don't call them patients, we call them subjects. So whenever I say subjects, I mean patients or participants in the research. So by following GCP, we're going to protect the rights, safety, well-being of our trial subjects. And secondly, what we'll do is we will ensure that any data that results from a clinical trial or from any research is complete, it's accurate, it's unbiased. So we have evidence for any claims we are making. So what I want you to understand and remember is, think of it in this way. By following GCP, GCP has two pillars. The first pillar is to protect the rights, safety, well-being of subjects. And the second pillar is making sure that any data which comes from our research, it's complete, it's accurate, and it's unbiased. So this is what we do. So this is the purpose of good clinical practice. How do we actually achieve this? That's the next question. If this is what good clinical practice is, how are we going to achieve this? Well, we achieve this through many ways. We achieve it through guidelines. We achieve this through the regulations we have in our countries, and we achieve this through laws. So guidelines and regulations. Who sets the regulations in our countries? These are our regulators. So for example, in Saudi Arabia, it's the Saudi FDA who'll be setting the regulations. So we have guidelines, we have regulations. How else can we achieve compliance? And we achieve compliance also through standardizing our processes. Let's all work to one gold standard and we follow it. So this is very important. Guidelines, regulations, and standards. All of these are now set up to ensure that the two pillars of good clinical practice are going to be followed. Okay, so now I'm going to say a little bit more about what these two pillars are and how we achieve them, because that is what we're going to do over the next 7 days, is how we are going to achieve these. But how we got to good clinical practice, I think we must understand how we got there. So what I'm going to do now is just give you a very brief history of good clinical practice, a brief history of research. So if we go back in time, so about 100 years ago in 1906, this is when we first saw any kind of regulation or guidance coming out that tells us this is how you should experiment in humans. So in the United States in 1906 saw the first type of act called the US Food Drug Act, which actually came out in the United States, and it was a simple act, and all it was saying was that if any food or drug supplies are being transferred across interstates, they must be not mislabeled, and it must be very clear what's contained in these products. A very simple kind of beginning. So talking about labeling a product. So how does this tie in now? Well, every drug has a label on it, yes? So this was the beginnings of what goes in a label. So that was like about over 100 years ago in 1906. Now, as we moved through time, something happened in 1937 where there was suddenly about 100 children died. And these 100 children died, and they were actually taking a medication. They were taking a drug called sulfanilamide, which was used to treat streptococcal infections. So throat infections, they were taking a drug and they died. Now, what actually happened here? Well, what happened, this drug, yes, was a drug, was working, but it was a very insoluble drug. And the only way this could be dissolved was in a chemical, in a product called diethylene glycol. Now, I'm not sure if you've heard of diethylene glycol. We use it a lot here. I don't think you need it in Saudi Arabia, but we use it a lot here in England, especially during the winter. It's what we call antifreeze. Yeah? We put antifreeze in our car radiators so our cars don't freeze up in the winter. It's a compound that was used to dissolve this sulfanilamide drug. Now, ethylene glycol is extremely toxic and it kills. And this killed all the children. And once this happened, in the US FDA produced the 1938 Food, Drug, and Cosmetic Act. And what this Food and Drug Cosmetic Act did, amongst a lot of things, it basically said you must be testing for safety of any products before you give them to human beings. And very importantly, get approval from the regulators before you want to start selling your drug. So again, the beginnings here of something we call the new drug application, which I will explain later on today. So 1938, another act came in. And also, as you know, during the 1930s, the war was on. The whole world was going through World War II. And I'm sure you've heard of the trials that were done by the Nazi Germans in Germany, and these were the 1947 physician trials. So what was happening in Germany in the prisoners of war camps, these prisoners were being experimented on. In the name of science, doctors were doing some very unethical experiments on these prisoners of war. And when the war finished, you had the Nuremberg trials. And from the Nuremberg trials, we saw one of our first documents which really gave some guidance on how to experiment in humans, called the Nuremberg Code. So in 1948, we saw the Nuremberg Code coming in. So just to illustrate to you what happened during the Nuremberg trials and how unethical the experiments were, I'll just give you a few examples. And these are real pictures which were taken by doctors who were researchers. And this is the research they were doing. For example, they wanted to see, how can we treat phosphorus burns? So what they did, they got the prisoners of war and put hot phosphor on their skins and let their skins burn, tied the prisoners down, let their skins burn, and see what happened. Very unethical. Another example of an unethical experiment they did, they exposed prisoners to ice cold water. So they put prisoners in ice cold water, and basically they wanted to see how long it takes for somebody to freeze to death in ice cold water. And from their experiments, they said, put them in ice cold water, it takes about 5 hours for these prisoners to die. All of this was being done in the name of science. Very, very unethical. Just 2 more examples. Here is another example. What they wanted to do was they wanted to see, what happens when there's continuous lack of oxygen. So at high altitudes, how long does it take for somebody to die? So they would tie up prisoners like this, do a little simulation and see what happens. And they documented everything. They took pictures and they documented. So for example, let's say they said at 4 minutes, the subjects begin to perspire or sweat, they wiggle their head. At 5 minutes, they're cramping. At 10 minutes, the breathing is getting very erratic. And 11 to 30 minutes, they die. All of these were done very unethically, without any scientific integrity, without any reasoning. But some of the worst type of trials that were done during the war in the prisoner camps were those which were done on children. And these were the trials which were done by a doctor called Joseph Mengele. You may have heard of him. He's very notorious, and he was known as the angel of death. And basically, he did a lot of experiments on children, amputated their limbs, live amputations, infected them with bacteria, transfused one blood from one twin to another twin, direct chloroform injections to the heart, and all sorts of horrible things like live autopsies, live dissections. They did all of these things. So sadly, this what happened in the war. The doctors were actually, some of them got away with this, some of them got punished, but this led to the 1948 Nuremberg Code. And this Nuremberg Code was now setting the foundations of, look, we understand we have to do research to progress our knowledge, but this research should be done ethically. And this, the Nuremberg Code was very short. It had 10 points in it. And the first one was voluntary informed consent must be taken. I'm not going to go through the rest, but just highlight voluntary consent. So there you go. So now we have this code, some guidance, which told us researchers as doctors that if we're going to experiment in humans, we must follow certain codes. Now, what happened as we progressed during the decade? In 1957, in Europe and the US, suddenly we saw children being born without limbs. And this was a thalidomide tragedy. And I don't need to explain that because everybody's aware of what happened with thalidomide. And this led to the development of an amendment called the Kefauver-Harris Amendment. And basically this started saying, look, you must do preclinical toxicological testing before you go into humans. So as you can see, our story is building up on how we started doing research, how all the laws coming in. So that came out. But what I really want to focus on very importantly is something very important, which is the Declaration of Helsinki. What happened is we understand we need to do research. A group of doctors got together and they started writing a document on how to conduct research in human subjects. And in 1964, these group of doctors, they called themselves the World Medical Association, and they produced the first version of the Declaration of Helsinki. The Declaration of Helsinki is one of the most important ethical documents for us researchers. It sets the ethical principles regarding human experimentation, and every researcher must read this. We all follow the Declaration of Helsinki. You are here on the webinar today because in the Declaration of Helsinki, it clearly states that every researcher must be trained in ethics and good clinical practice. So this document, the Declaration of Helsinki, came out in 1964. That was the first version, and this is the cornerstone of human research ethics. And this document must be read by everyone. So as a takeaway message from this evening's seminar is download the Declaration of Helsinki. I shall tell you where you get it from and read it. So as I mentioned, the first version came out in 1964. However, as we progressed with research, our knowledge gained, we needed to update the Declaration of Helsinki. And we updated it with new clauses in there, new information was put in there. And the latest version we are following at the moment is the Declaration of Helsinki 2013 version. So that is the current version. All of us researchers, like yourselves, like pharmaceutical company, government organizations, we all, whenever we do research, we quote, we are following the principles in the Declaration of Helsinki. So the Declaration of Helsinki, again, I'll reiterate, very important. Please take a moment to read this. It's a small document, about 8 pages only, and you can download this directly from the World Medical Association website, and I've given you the URL on this slide over here. So Declaration of Helsinki, very, very important. So now we have, as we progress, 1960s, we have the Declaration of Helsinki coming along. Then what happened in 1970s in the US, the Tuskegee study, syphilis study was displayed, was released to the world. What was happening is in the US government was in fact doing a study on syphilis in the Black African American population. These subjects were in a placebo-controlled study. Well, it was a placebo study, and they wanted to see what happens to the progress of syphilis. The subjects were given a tablet, which is placebo. They didn't know they were in a clinical trial, but one doctor found out that this was very unethical. This was raised up, and what happened in the United States, they released the 1979 Belmont Report. And this Belmont Report is a little bit similar to the Declaration of Helsinki, but I won't say a lot about this at this stage. So that's good. So 1970s, we're coming closer to time. Then as we started doing more research around the world, every country started to develop their regulations. So for example, the FDA produced their guidelines. The UK produced their guidelines on how to do clinical research. The French produced their guidelines on how to do clinical research. So the situation was that if I wanted to do a research in England or register a drug for sale in England, I needed to follow the UK regulations. Now, the same drug, if I wanted to get the registration available, marketed in the United States, I needed to follow the US regulations. So do the research again, but under the US regulations. If I go to another country, they said, can you do it under our regulations? So this, as you see, didn't seem to be the best way of getting drugs to the market. Yeah? So what was happening is everybody was doing something slightly differently. And what happened in the 1990s, the world bodies got together. So the world researchers, the world governments got together and they said, look, we all want to do ethical research. We want to make sure that when we do research, it's ethical, it has scientific integrity, and very importantly, we all do it to a highest level. So why don't we, so the European Union, the Japanese, and the US FDA regulators got together and they said, look, why don't we all work together and decide and agree on a specific standard that if I do a clinical trial anywhere in the world, then that trial is going to be very ethical and it's a standard that we can all accept. So what happened is the regulators, so the European Union, the US FDA, the Japanese, why it was these three regions is because that's where traditionally most of the research was being done. They got together and they had other people, they had the World Health Organization inputting into this. They had the Nordic countries inputting, everybody was inputting, and they developed a set of guidelines. And these guidelines were called the International Guidelines, the International Council of Harmonization, or the ICH Guidelines. So the ICH Guidelines on Good Clinical Practice, we call ICH GCP. So what we saw in 1996 was the birth of these international guidelines, which everything I am saying to you today comes from these international guidelines. We saw the birth of Good Clinical Practice, or ICH GCP. So what does ICH GCP stand for? Well, this is the direct translation from ICH GCP, the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. So this is what ICH is. So ICH is an organization now which is producing a lot of guidelines for the international community. Whenever we're going to do research, we all agree to follow these guidelines. So since 1996, which is about 25 odd years ago, we are all in the world, whenever we do research, we follow the same standard. So whatever I do in Saudi Arabia, which is being done now, we are following the same guidelines which are being used in the UK, being in the US, in Swaziland, in Australia, everywhere. So the objective of these guidelines is very importantly to provide a unified standard for the European Union, Japan, and the US to facilitate acceptance of clinical data by the regulatory authorities. So for example, if I do a big clinical research program in the European Union and I want to register the data in European Union, the US FDA should say, you know what, that research was done to such a high standard because we follow the same standard, we will accept that data. That is the purpose of this, to make sure we are following good ethical guidelines, but very importantly, ensuring that we are bringing drugs, new drugs to our patients sooner. So when this guideline was being developed, we did have input, as I mentioned, from other bodies such as the World Health Organization, Australia, Canada, and other countries. So this is what ICH is. And one specific guideline within ICH is called GCP, and that's good clinical practice. And that is what we all focus on as researchers. So I just said there's one specific guideline. Well, what happens is ICH has produced many, many guidelines. And what they have done, they've put them in different categories. And these categories are called the E, the S, the Q, and the M categories. And the ones we are very interested in are the E categories, which are the efficacy categories. So if I go into the efficacy category, I will see many, many guidelines there. All these guidelines tell us how to conduct research. And one guideline, which is the E6 guideline, that is good clinical practice. So for the next 7 lectures, everything I am saying is coming from these E6 good clinical practice guidelines. And this is what you as researchers are expected to follow when you're going to do research in human subjects. And this is what it is, E6. And the E6 guideline, this is their official name, is the Integrated Addendum to ICH E6R1 guideline for good clinical practice. As I mentioned, they came out in 1996, but in 2016, they were updated, hence we have revision 2. ICH GCP E6R2 are the good clinical practice guidelines. And these are the guidelines all researchers live and breathe every day. Now, these guidelines, if I look at the E6 guidelines, these guidelines, you can get them, download them from the ICH website, and I'll show you the URL later. So if I look at these ICH GCP guidelines, they have a number of sections in them. Section 1 all the way down to Section 8. And what these guidelines are actually doing is telling everybody what you need to do. So they identify 3 very important people who are involved in research. One are the Institutional Review Board or the Independent Ethics Committees. And tomorrow I shall tell you what they are saying about these IRBs or IEC. Then they talk about the investigator. And this is what a researcher is. So the kind of standard international term for a researcher is investigator. So from now on, I'll use the term investigators. So yourselves as investigators, you have to read very carefully what is in Section 4 of the GCP guidelines. So Section 4 is the GCP guidelines. And this tells you if tomorrow you're going to do research in a human subject, this is how you should do it. And the third person it talks about is the sponsor. So this sponsor is the individual who is the sponsor of the research. And in broad terms, this could be, for example, the pharmaceutical company, the biotechnology company, a big non-NGO, or whoever has got the product and they are responsible for the financing of this product. They are the sponsors. And then you have another number of different sections. So for example, it tells you how to write a research, what should be inside a research proposal, what we call a clinical trial protocol. So we have a protocol. So every research is going to have a protocol. What do I put in the protocol? Section 6 of the GCP guidelines tells you this is what you put inside it. So every company around the world who's writing a clinical research protocol is following exactly what's in Section 6. Then you have something called the investigator's brochure. And I'll say more about this in a very short while, what the investigator's brochure is. And the final section of the GCP guidelines is Section 8, which talks about essential documents. And one of our webinars is on essential documents, and I'll explain what these essential documents actually are. Okay. So looking at this, so going back, section 1 is a glossary. Section 2 talks about the principles of good clinical practice. And what I want to do is take you through these principles. What do they mean, and how are these going to impact us as researchers? Now, in section 2 of the GCP guidelines, we have 13 principles of GCP. And we are now going to talk about these 13 principles. So the first principle is that clinical trials should be conducted in accordance with the ethical principles that have their origin in the Declaration of Helsinki and that are consistent with GCP and the applicable regulatory requirements. As I mentioned earlier, the Declaration of Helsinki is our foundation of doing research. So the GCP guidelines actually default to them as well. These guidelines are building on top of the principles of Declaration of Helsinki. So everything in good clinical practice is in line with the Declaration of Helsinki, but is providing you some more guidance. It's a guideline. So this is very important. And even if you look at the Saudi FDA regulations, they're clearly quoting there, we follow the declaration of the principles in the Declaration of Helsinki. Therefore, very important, you do take a few moments to read this document. So that is the first principle. The second principle, here it states, is before a trial is initiated, foreseeable risks and inconveniences should be weighed against the anticipated benefit for the individual trial subject and society. A trial should be initiated and continued only if the benefits justify the risk. We all know that there are risks in doing research, but this is clearly saying you have to look at the risk benefit of doing any research, and the benefits should outweigh the risks. This is what it's saying. So that's very important. So in every research, in our protocol, in our proposal, we need to clearly place a risk benefit statement. What are the risks? What are the benefits of doing research? So those of you who are writing research proposals or protocols, think about this, because this is something, it's a principle of good clinical practice. Next principle. The rights, the safety, and well-being of trial subjects are the most important considerations and should prevail over interests of science and society. This is very important. This is saying you always put subjects first. In research, the most important person is the subject, and they are the priority. You cannot put your interests of science over the safety and the rights of the subjects. If we go back and look at the pillar, one of the pillars of GCP is to protect the rights, the safety, and well-being of clinical trial subjects. So take-home message over here, subjects, we must make sure they're protected in our research, and we cannot do anything that'll compromise their rights, safety, well-being. The next bullet is, the next principle is that the available non-clinical and clinical information on an investigational product should be adequate to support the proposed clinical trial. The term investigational product is the international term for the study drug or the study medication, what is actually being tested. So we call it investigational product. And one of the lectures I'm going to talk about, one of the webinars later on this week, is on investigational product. So what is this, the available non-clinical and clinical information? This kind of information is contained in a document, and this is the document which we call the investigator's brochure. So what happens is, let's say there's a drug which is under development and it is not yet on the market. It is still being tested. It's in the research field. It's in phase 1, phase 2, phase 3 type of trials. And I'll explain these phases to you very shortly as well. Any data about this product must be presented in a document, and that document we call the investigator's brochure. So GCP tells you what goes inside an investigational brochure as well. The next principle is the clinical trials should be scientifically sound and described in a clear, detailed protocol. So very important here, every research has to have a protocol. It's a principle of GCP. So there has to be a protocol telling what you're going to do in this research, what goes inside the protocol. The GCP guidelines tell you what goes in trials. It's one of the sections in GCP. The next one is a trial should be conducted in compliance with the protocol that has received prior institutional review board, IRB, or independent ethics committee, IEC, a favorable approval or opinion. Now, this is very important. So basically, we cannot conduct any research without getting approval from our IRBs. It's a principle of research. So every research has to be reviewed by an independent body, which we call an institutional review board, who will look at the science of the proposal, they will look at the ethics of the proposal, and they will give you the approval. And tomorrow, I'll tell you more about IRBs and IECs. So another very important principle of GCP. Another principle here is that the medical care that is given to and the medical decisions made on behalf of subjects should always be the responsibility of a qualified physician or a dentist if it's a dental type of research. So what this is saying is in our research, there must be some medical input. Who is going to be looking after these subjects in a clinical, in the research? We go back to the risk-benefit assessment statement, what we have, and you need to tie it in with how will the subjects be protected, and they must be protected by a medically qualified physician. A very important point over here is each individual conducting a clinical trial must be qualified by education, training, and experience to perform their tasks. So as researchers, if you're going to be conducting a clinical trial, you must be qualified by education, which you are, training and experience. Now, for example, some of us may be epidemiologists, oncologists, or whatever. We're very experts in our fields, but how much do you know about conducting clinical research? What do you know about good clinical practice? So basically what this is saying, you must have GCP type of training to conduct research. And this is what we are providing to you over this next set of webinars. Is this GCP training, is to meet this point of the principles of GCP. Look, we are training our researchers on good clinical practice. Very important point. The next point here, and this is freely given informed consent should be obtained from every subject prior to the clinical trial participation. This, if you go note back, was the first bullet point in the Nuremberg Code, is freely given informed consent. This is a very, very sensitive, very, very critical aspect, because now you're protecting the rights of the subject. Every subject has to have the autonomy to decide whether or not to participate in a research. And this is extremely important, and I shall be doing a webinar purely on informed consent, is how should informed consent be taken, how should it be documented, and what is the process we will do. So informed consent is critical. This is fundamental of doing research. Yes, one of our gateways to start research is we have to have IRB, IEC approval. That's a must. Once they've approved the study, before any subjects should be enter a clinical trial, they must give written, documented, informed consent. That is their agreement to participate in the research. This is a principle of good clinical practice. Another aspect here is that it says all clinical trial information should be recorded, handled, and stored in a way that allows its accurate reporting, interpretation, and verification. This principle applies to all records referenced in the guideline, irrespective of the type of media used. So what does this actually mean here? All trial information should be recorded, handled, and stored that allows accurate reporting, interpretation, and verification. So this is looking at the data. So this is now looking at the second pillar of GCP, which is ensuring that data is complete, the data is accurate, and it is unbiased. So one big difference of doing research from medical practice, you will find, is the volume of documentation that is required. Everything you do in research has to be documented because the principle of GCP is saying all clinical trial information should be recorded, handled, and stored that allows its verification. So for example, what this means is if you do some research and you have data and you're saying, look, this is my data and my data is telling me this, we should be able to verify that data. Where did you get this data from? Where was it documented? We want to be able to verify that data, and that's something which we call source data, which is part of essential documents that I shall be talking about later on this week. So very important here is looking at the data now. Next principle is the confidentiality. Now, again, we're protecting the rights of the subjects. The confidentiality of the records that identify subjects should be protected. So anyone who is in a research, we must protect their identity. Their privacy, confidentiality has to be protected. And I'll say more about that during our essential document source documentation webinar. So this is very crucial because there's a lot of laws and regulations in every country which talk about data privacy. For example, last year, I'm sure you're aware that in the European Union, we had the GDPR come out, talking about protecting the data subjects' anonymity, making sure that their rights are protected, their privacy, confidentiality is maintained. So when a subject enters our clinical research, we need to give them assurance that all of their data is going to be protected, their identity is going to be protected. The 12th bullet says investigational products should be manufactured, handled, and stored in accordance with good manufacturing practice. And they should be used in accordance with the approved protocol. Now we see a new term here called GMP. GMP is good manufacturing practices. So what this is saying is to our, for example, sponsors who develop the drugs, who make the drugs, when you're making the drug, you have to follow certain guidelines, regulations that they're manufactured, handled in accordance with GMP. What we do is GCP, good clinical practice. So GMP doesn't really affect us. It's in the manufacturing aspect of the whole drug process. So I mentioned to you earlier, ICH produced ESQM guidelines. One of the guidelines in the Q guidelines talks about good manufacturing practices there. So there you go. So all our products are pure, they're labeled correctly, everything. That is what GMP is saying. So the final principle talks about systems with procedures that ensure quality of every aspect of the trial should be implemented. Aspects of the trial are essential to ensure subject protection and reliability of trial results and should be the focus of systems. To summarize that, what we're saying here is we want quality in our clinical trials. Every data point we have should be good, true quality data. There should be systems put in place, checks, for example, that ensure that our data is accurate, it is complete, and we are producing quality. So if you go back to the definition of GCP, it was an ethical, scientific quality standard. So quality is very, very important to us. So these, for example, are the principles of GCP, the fundamental aspects we have to follow what GCP is asking us to do. I mentioned to you that the ICH talks about 3 different players: the IRBs, the investigators or the researchers, yourselves, and the sponsors. All of us have got obligations. As researchers, our obligation obligations are identified in section 4 of the GCP guidelines, and I will be focusing on these obligations over our series of webinars. If we follow our obligations, we have followed good clinical practice. And if we followed good clinical practice, then we have followed the international requirement, the international standard that is acceptable around the world of performing research, performing good clinical research. So that is what, as an introduction, what GCP is, what the principles of GCP are. So the next question is, we have all of these principles, and I have just given you an introduction now, and we are going to build onto each one of these in a lot more detail as we move along. But what I'll do now is explain to you, give you a little overview of the drug development process, how GCP is coming in here, and very importantly, how does this tie in with the Vision 2030 of Saudi Arabia of doing more research? So what I'm going to do now is tell you, if today we find in the laboratory a potential cure, a potential molecule which we need to develop, how do we take this through the drug development process up to the market is a process. And this process is a long process. And this process I'm now going to explain to you. So I'll give you a clue, it's a long process. Okay? So the road is very long. So what we can do is we can kind of summarize this, the drug development, as 4 different kind of stages. You have a drug discovery stage, a preclinical stage, and the clinical trials or the research stage. And then finally, the regulatory review stage. And this is what happens during each stage. So let's say what happens, we find we've got a disease, we've seen a receptor, for example, and we develop a molecule. So during discovery, our biochemists, our pharmacologists are busy, our computer technologists are busy looking at receptor pharmacology, and they're churning out thousands and thousands of new molecules. Yeah? So thousands of molecules are being produced. And some of these molecules may look promising, and they will then take them into preclinical. So now we go with a few molecules into the preclinical testing. And when we say preclinical testing, this is the testing on animals. So this is where the animal testing will come in. So the in vitro testing, the in vivo testing, looking at animal models, looking at diseases in animals and seeing, is this molecule actually working? What is it doing? Is it safe? So we test lots of these compounds. And let's say, for example, we see some potentially good compounds which are showing promise in animal models that, yes, this drug looks like it's working. So our next stage is we have to test this on humans. Yes? So this is, we're now going into human research. Okay? So before we can go into human research, I want to introduce a new term to you, a very important action has to be taken. Before we can go and test our drugs in humans, we want our regulators, our governments, our Saudi FDA to give us approval to go and test this in humans. And what we do is when we make a submission to our regulators, we tell them, look, in a submission with documents and everything, this submission is called an IND or an investigational new drug. So we do an IND for our clinical trial. So IND, investigational new drug, is submitted to our regulators. So for example, if you want to do research in Saudi Arabia, a clinical trial with a compound which is not on the market yet, we will submit to our Saudi FDA. So this is the process for drugs which are not on the market, yeah, which are under experimentation. So it goes to our regulators, and our regulators either say yes or no. You can do the trial or you can't do the trial. So let's say, for example, they say, yes, go ahead, you can do your clinical trial. Before we can do any research in humans, the principle of GCP says you must have IRB approval. We will submit to our IRBs and ask for approval to do the study. So to do research in experimental investigational products which are not on the market, we need 2 approvals, our regulators and our ethics approvals. So let's say our regulators say, yes, you can conduct the research. Our IRBs say, yes, you can conduct the research. then we follow a system of how to do research in humans. We do our clinical trials. And these clinical trials are divided into 4 broad phases. We have phase 1 of research, phase 2 of clinical research, and phase 3 of clinical research. In each one of these phases, we are doing something different. And I'm going to explain these phases to you very, very shortly. So we do our research, phase 1, phase 2, phase 3 research. So we're testing this drug in humans. We're seeing, does it work? Is it effective? Is it safe? And let's say, for example, this drug is looking very good. It's looking very safe. What we want to do next, or pharmaceutical companies, whoever, they want to register this drug. They want to be able to sell this drug. They want this drug to be made available in the market. So what we do is, to get that license to sell the drug, we need our regulators or our Saudi FDA to give us the approval to do that. So what the pharmaceutical drug companies, biotechnology companies do, they will submit a big dossier, a big package of a lot of data which summarize all the clinical research, all the trials in a lot of detail, the preclinical data. All of this goes in a package, a dossier, which is sometimes called the NDA, or the New Drug Application. So NDA, New Drug Application. It's in that, this is in a lot of countries, it's known as the NDA. In Europe, we call it the MAA, the Marketing Authorization Application. This is then submitted to our regulators. Our regulators will then review all the data, and they will look, is this data correct? Is it accurate? Does it answer the right questions? Do we need this drug? Does it work? And if they say, yes, we've given you approval, you can go and then license and sell the drug. The drug is then on the market. So the drug is then on the market and it is available. Does clinical research finish there? No, it continues. So any research that is then done on that product is what we call phase 4 clinical research. So this is what phase 4 is. So phase 4 is research on any product that is on the market, but we're researching it. Okay? And under phase 4 research, this is where we get a lot of what we call the non-interventional studies are coming in. The observational type of studies are coming in. This is phase 4 research. So this is actually a brief summary of research. Now, how long does it take, for example, to get one drug from, if I find it in the labs today, to get it on the market? How long does it take? It's a long process. Approximately, you're looking at 12 years, 12 to 15 years. It's a long, long time. Now you say, oh my God, this is crazy. It's a long time, but for example, we've got COVID at the moment and we can't wait 15 years for anything new from COVID, any new drug 15 years, we need it now. What can happen is we can reduce this timeline. We can look at a lot of ways on how we can get drugs to the market sooner. We can look at what type of phases we can use to register a product. So bottom line is it's a long process, a lot of people involved, and it's an expensive process. So just to give you an example, to get one molecule through this entire drug development process from discovery to the market, it costs in the region of 1.5 to 2 billion US dollars. It's a very, very expensive process to get a drug on the market. So take home, long process. Now, important is the clinical trials. All of these clinical trials, phase 1, all the way 1 to phase 4, must be conducted according to ICH GCP or to the good clinical practice guidelines. So just to very briefly summarize to you, and in case you're not aware, what do we mean by phase 1, phase 2, phase 3, and phase 4? I will explain what these trials are. So the phase 1 trials, these are the phase trials which we call first time in humans, or the FTIH trials. So this is where we go first time in humans and we're going to test the drug. We've never done it in humans before, and it's the first time. These are very high-risk clinical trials, and these trials are conducted by specialized units, or they're conducted in hospitals where the volunteers will come into the hospital or the unit, they will be dosed and they will be observed. And what we're looking over here, very importantly, it's a very exciting phase, is what does this drug actually do? What is the safety of this drug? And very importantly, are there any indications it will work? Usually our phase 1 trials are done in healthy volunteers. And the reason we use healthy volunteers is if, for example, we give them a drug and an event happens, we know that this event is due to the drug and maybe not any underlying condition. So it's usually in healthy volunteers, but not always, because sometimes it's unethical to go in healthy volunteers. For example, if they're oncology products, which are very toxic, we will not give them to healthy volunteers. We'll go straight into patients with the disease. So phase 1 trials are usually very small trials, about 16 to 20 per trial and about 100 in total. And we look at lots of things. We look at the pharmacokinetics of the drug, the bioavailability of the drug. And through this data, we will know what the dosing regimen may be of the drug. What is the half-life of the drug? But our primary objective of phase 1 trials is this drug actually safe? So let's say we do our phase 1 clinical trials and the drug is looking safe. We have the science behind it that it should work, and maybe we have some evidence from our phase 1 trials it works. We then go into the next phase of research, and we call this the phase 2 of clinical trials. And this is the first time we go into patients. Again, very exciting phase 2 trials, because now we're going to see, does this drug actually work? At the moment, we don't have any idea on what the dosing should be, what the right dose is. So we do dose ranging studies, for example. We'll do we'll see what dose is the maximum tolerated dose, for example. And we look at efficacy. Does this drug work? Is there an indication? And is it safe? If it's not working, it's not safe, drug development stops over here. But let's say, for example, these trials are fairly big trials, not very big trials, but about 200 to 300 patients trials, maybe 1 or 2 or 3 trials, depends on how we want to develop this. We then, let's say the drug is looking very good. We have an idea that, yes, this is the right dose. Then we need to confirm this. And this confirmation is provided in phase 3 of clinical trials. So phase 3 of clinical trials, this is where we say, we have a drug now. We are going to compare it with a gold standard. What is the standard best treatment, and see head to head, how good is this drug? So these are our confirmation of efficacy trials. These are very, very big trials, international clinical trials, very expensive trials. If you look at the costs coming up over here, and these are known, we include patients from all over the world, different ethnicities, and include them and get the data to prove the drug works. And these phase 2, these trials will give us our evidence that yes, the drug works, but very importantly, all of these trials should have been conducted to ICH GCP requirements. So that's phase 3. So once phase 3 is done, the drug's on the market, we have our phase 4 clinical trials. And these are the trials which is when the drug is on the market and we want to explore. We want to get more information. We look at new therapeutic opportunities. We look at observational studies. We look at real world evidence over here. How is this being used in real world? What are the safety signals? What are we getting? Yeah. And these are phase 4 trials. So this is a little bit of a summary for your phase 1 to phase 4 trials. Now, at the moment, in the kingdom, of Saudi Arabia, we are doing trials, mainly a lot of phase 4 trials, a lot of observational types of trials. But we need to be doing a lot more of these phase 2, phase 3 type of trials, the really exciting trials. They are high risk, the trials, but these trials are very important because they bring new medicines, new opportunities for patients into the countries to allow them to go on some type of treatment. So that is, in a little bit of a nutshell, a little overview of phase 1 all the way to phase 4. Very, very important. The vision of 2030 is to do a lot more research. We want to be international players in research and bringing more of this research into the country. The only way we can do more research in the country is to actually ensure that when research is being conducted in any institution in the kingdom, this research is being conducted according to ICH GCP. That is what we need to demonstrate. We need to train everybody so they understand the requirements of GCP. So I said I'd speak for an hour. I think I've spoken for an hour. So what I'd like to do now is open up the floor for any questions and answers. So Dr. Tariq, I'll hand this over.
Yes. Thank you so much, Mr. Naeem, for this exciting presentation. I hope all the audience captured all these important informations. And now we can take some questions. We received actually some are long, some are short. But anyway, I wish to inform the audience that everyone can feel free to post his question right away using Twitter hashtag research_Saudi Vision 2030, and we will take them in order after we take the ones that we have already received. If the time frame did not permit, the presenter will kindly answer the rest of them later in Twitter. So the first one, we have it here, and he mentioned asking,